Be prepared for what you are going to get from the public. If you are not prepared with some kind of messaging when you find it in your state, if you're not prepared to have a system to receive reports, you are going to be inundated, especially depending on the population area where you find the pest. We were receiving thousands of reports every week. Our staff could not keep up with that. We worked with Penn State. We developed a Survey 123 reporting mechanism on our webpage. I know that New York has also done the same. So you have people who know how to do this. I'm not the person to ask, but I will find the person for you in Pennsylvania. Or we also have New York here. If you have access to Survey 123, use it, okay? Consider it a canine team. They're fabulous. Lucky, we have her. Shane is the handler. He does wonders, takes her out to businesses. She helps educate those businesses on where to look, and the businesses appreciate it. She's a great outreach mechanism. She even has a book that's been written by Extension for her. So we're really excited to have her, and we're going to be adding another canine team. We've got Ruby, who's in training right now to join us, and she'll be on the west side of the state. Okay. How are you going to work with all those industries? I'm going to give a talk a little bit later about who do you tell? Well, you have to find a way to make it work for you. The permit system is what works for Pennsylvania because the businesses go through that training. They're told what to look for, when to look for it, and what to do when they find it. 35,000 companies have gone through that training. And we ask them how many permits they're issuing, how many people they're training. And it is over 1.3 million people have been trained on spotted lanternfly through that process. Again, our partnership with Penn State made that possible. Okay. Biggest thing, think outside the box. This insect does different things. Julie will look at me and go, I've never seen that before, Dana. I don't know why it's doing it here, but I've never seen that before. And she's the authority on them, okay? So your partnership with Penn State, with USDA, they're critical. Also your sister agencies, DCNR was right there with us, with Ho Ping, to start research, to see what he could do. Your industry groups like Hardwoods, local governments, as the secretary mentioned, and then other universities, Temple. You know, Matt, I can't say enough. He's done some modeling for us. He's continued to work with us. We just value all of those. So we've learned a lot, still have a lot to learn, still questions to answer, but we're going to do it together because you will not do this by yourself. If you don't have extension, if you don't have your land grant university and USDA, you're not going to make it. So please do that. And just know we're here. Pennsylvania is always happy to answer questions. If you want to come work with us, we've had states work hand in hand with our operations teams during the season. They'll help you. We'd love to have you. We'll teach you everything we can. So right now I'm going to turn it over to Matt Travis. He is the National Policy Manager for Spotted Lanternfly with PPQ USDA.
Thanks, Dana. Yeah, good morning. It's great to see everybody. It's great to be back here and face-to-face. It's also great that we could do this hybrid. And so we have a lot of virtual participants as well as, you know, in-person conversations, discussions, you know, and I'd really like to thank everybody for making the trip. I know, you know, budgets what they are, timing of the year, you know, puts all those challenges on us. And so traveling here is very important. And certainly I'm glad to see that there are so many still committed and still pushing forward on learning more about this pest. And this is what this kind of meeting is about. The summit is about learning. It's about learning from each other, learning about each other's experiences. And like Dana said, it really is about the team and the relationships. We could not get as far as we have without the relationships, without the partnerships, working together, federal at the USDA APHIS level, state, municipal, extension, academia, all valuable members of the team. All the industry partners, very, very valuable as members of the team. And so it's just great that we're able to get together and learn from each other and learn from each other's experiences and just continue to understand more and more about this pest, hopefully putting it to good use to limit the advancement of the spotted lanternfly. Spotted lanternfly is a complex pest, right? It does different things. And so, you know, again, getting together and learning from each other, supporting research, limiting the movement of the spotted lanternfly, and then working on a consistent outreach message is really critical. And Dana mentioned the strategic plan. You know, it was a great opportunity to work together with the National Plant Board, with NASDA, to be able to put together a 5-year strategy for this invasive pest, because it just laid the guidelines and the groundwork for us to go forward. And I would say that that strategic plan can be applied to a lot of different pests. So we certainly looked at other pests and compared as we went out on our building the plan together. So the relationships with all those organizations made that happen. And it's now time that we start to implement that strategic plan as we work towards implementing it. And later today, I'll be talking more in depth about the strategic plan and how we can implement it. It is very valuable for states that have spotted lanternfly, as well as organizations and states that haven't seen it yet and haven't detected it yet. And so that plan really lays out a foundational guideline and groundwork to help us move forward and hopefully limit the advancement, give us time for science to continue to build solutions and solutions, long-term solutions like biocontrol, which is a very, viable solution that USDA APHIS is committed to, and we're working very hard on along with other partners at the university and other federal agency level. We continue to look for solutions that we know are out there. And as the secretary said, we know that it's through sound science that we're going to get there, and it's building that team to work towards that goal. Again, I just want to say that as we focus our conversations or discussions in the next day or two, that we really listen and we really hear and understand the experiences that people will be sharing, and that we take those back. We take those back to our wherever we call home, and we work with our teams back at home, taking that knowledge, that understanding, but also know that, just as Dana said, we will still always be here. We'll be here to have those conversations and discussions. We consistently meet every week to talk about spotted lanternfly, both with a cross-functional working group within APHIS, but also we talk to our state partners, our academic partners, and we put together plans for research and operations consistently all the time. So we're still here. We're a resource, and certainly Pennsylvania has been a leader in this. And so we're really happy to work with states like PA and all the other states to continue this program and to really learn and meet the challenge of spotted lanternfly. Again, I know that we can make a difference. I know that we can have successful outcomes. And I really look forward to learning more on how we can safeguard, American agriculture and certainly facilitate trade, because that's critical and important for all partners involved here. Again, thank you, everybody. Thank you for the opportunity. And I'm glad to see everybody here. I'll turn it over to Julie Urban. Thank you.
Good morning. Thanks everyone for being here, and thanks for the opportunity for me to be up here. As many of you know, I'm a researcher working on spotted lanternfly. I was called in day one, September 22nd, 2014, man. And so I had the opportunity to come to Penn State 2.5 years later. And so basically I'm the cat herder of the researchers is basically functionally my job. Okay. And so kind of pulling everything together, what you've heard today already from the great introductions that Secretary Redding, Dana, and Matt gave, is that two things kind of emerged to me as being really important for our experience with spotted lanternfly up until now and moving forward. And one is the importance of speaking and interacting across silos. So like in a research perspective, you know, we know the importance of interdisciplinary research. And certainly that's important. That's what we've been doing, but this is bigger than that. Okay. This is the importance of researchers talking with regulatory folks. As Dana already expressed, they're like the front end interacting with industry and government and all these groups like, okay, but I don't have your job. Okay. But basically, you know, have the regulators talking with the researchers, talking with the extension educators, and having like that constant dialogue. And that's what we're doing here today, right? And so what's really amazing to me is that, Dana, if you saw it coming or not, but basically this SLF Summit has provided us with a framework to have that dialogue and to continue to do that moving forward, even as different individuals transition in and out of our teams, right? And so that's really cool. The other thing that folks have alluded to is that as lanternfly spreads, it's always throwing us a curveball, always something different. That's partly as it expands to new areas, but also it just is contacting other industries, businesses, and whatnot. And so with that, what's been really neat for me to see is that brings more people into the dialogue, right? More people to talk about their problems. And again, the format of this summit allows us to do it. We're hearing about new states, for example, you know, Tennessee, Illinois, Kentucky here. We're talking about now that lanternfly is getting close to the Erie grape growing region. There's differences in terms of how grapes are grown there, you know, with cooperatives and with automated harvesting and different problems we haven't experienced yet. So this is allowing us the opportunity to talk about those problems and deal with them. And so to me, I kind of see this as like, we have this emergent collaborative leadership that continues to evolve on spotted lanternfly to deal with all of these problems. And so I think that has been built, so that's going to continue to expand. And I think that that's going to be our best bet for moving through this and dealing with spotted lanternfly as well as we can. So with that, I think I'd like to kick off the first talk. And so our first speaker is Dr. Dennis Calvin, also an entomologist from Penn State. He's currently retired, but he served for 9 years as Associate Dean and Director of Cooperative Extension. And then after that, 2-3 more years, well, he's still doing stuff with us now, as the Associate Dean of Special Programs, where he was leading up our work on spotted lanternfly. And now he's a groupie for his grandson's baseball team, but he still managed to publish a really huge paper that he's going to share with us. So here, welcome, Dennis.
Well, even though I'm retired, I can still make that step up there anyway. So that's a good thing anyway. But I thank you, everybody, for inviting me here today. And good morning to everybody. What I'd like to do is introduce this first talk. It's basically the spotted lanternfly in Southeast Pennsylvania, and we're going to focus on predicting key life stages over the course of the year, the seasonal development or activity of spotted lanternfly. But I'd also like to point you to the citation there under the title. This is the paper that this work came out of. So if you want to get more detail or get the actual degree days that the title of this presentation says, they're in that paper. I'm not necessarily going to show you those today, but you can get them off of there. And I'm going to focus on some of the other aspects of this paper. I also wanted to mention that there's the team that was involved in this work, and Julie was one of those. Brian over here was one of those individuals. And I don't know if there's anybody else in here as well, but it was a group of people that did this work in 2019 and 2020. So now the tough part after being retired is figuring this thing out here to make sure I know how to change the slides here. So I guess the one in the middle. Nope, that's the light. Okay, is that it? The green one in the middle. Okay, there we go. So now I've got the technology down. We'll move on from here. But anyway, so why is this work important? Why is it important to be able to see when these pests are active during the growing season out there across the landscape? And well, some of the reasons for that are it's better time key management activities, things such as trap monitoring, scouting to estimate the size of the population in a given area, make better management decisions. If you're running a surveillance program, knowing where and when to run those programs. If you're looking at biological control releases or timing of when you might be out there detecting these individuals, it helps with that. If you're trying to put a pesticide application, you want to time it appropriately to get the biggest effect from that material that you're putting out there. And then finally, it gives us a mechanism to evaluate where the geographic distribution of this pest may or may not extend into as time goes on. So specifically today, some of the goals of this presentation are really to clarify what I mean when I say the activity period. And we'll have a slide in here. We talk about that in a few minutes, but also to illustrate the differences that we saw with spotted lanternfly relative to the activity period when they were on Tree of Heaven or when they were on red maple. And both of these are season-long hosts for spotted lanternfly with all stages showing up on both of these tree species. And then to discuss the activity period and how degree days influence the timing of these, as well as potentially other environmental cues such as day length. And then look at and make an argument for why, when we get to the adult stage and egg laying, why we need to reset that degree day timer a little bit later to the first adult in order to better predict the timing of those later events in the growing season. And then lastly, show you some slides that show what we think the geographic distribution of this pest could become or currently is across the landscape. And so just to talk a little bit about some of the methodology that we used for this pest, we actually looked at 20 trees in 2019. 19 of those were red maple, one was a pin oak. Don't ask me why we picked a pin oak, but we did. So we kept with those trees as time went on. And then 2020, we used the same trees, but we also added 3 ailanthus trees that we were monitoring just to see, you know, how things went on those trees relative to the red maples that were nearby. And then to monitor those trees, we put sticky traps on them at about chest high, which was 1.37 meters above ground level approximately. And those were 20.3 centimeters wide. And we wrapped those around the trees, and we'll show you a slide of that. But we put a wire mesh over, of course, to keep the bycatch out of there. That should be counted instead of counter the number of spotted lanternflies on 20 trees. We did that weekly over the course of the season from May 18th in 2019 to from June 1st in 2020 to the end of the season, which was really early November. And both of those years, we had a hard frost that killed everything at that time. And so we were pretty much done at that time of years. But we replaced those traps as they became a little bit saturated or every other week just to make sure they didn't become saturated and we weren't getting a good estimate. We actually conducted a second study that at the time we didn't know was going to be part of this particular work, but we just wanted to track spring egg hatch to verify the data that Erica Schmeyers had gotten and heard in terms of the rate of egg hatch in the spring of the year. So we did that study on 20 other trees, or on about 100 egg masses out there on trees that were nearby where we were studying. So they were really in the same area, and followed that from April 20th to June 18th when the last egg match hatched. As far as adults were concerned, we caught those on the trap. We also counted everything below them on the tree trunk and anything that might be crawling toward the tree that was down in the grass or something. And as far as eggs, we just looked up in the tree and we counted what we could see as far as we could see up. And these were 30, 40 foot tall trees. So we could see most of the way to the top to see where those egg masses were. Just to give you a sense of the kinds of numbers that we measured or we counted out there during the course of the season, we had 2019 on red maple, 2020 on red maple, and then on ailanthus. You can see in 2019, we started out with just around 5,000 spotted lanternfly. By the time we were to the 4th instar, we were down to 347. In 2020, we got a little bit earlier start in the hatch period. We counted 85,000 first instar spotted lanternfly. But by 4th instar, we were down to 506 nymphs that we were counting over that course of that time. And for ailanthus, we started at 683 on those 3 trees. If you prorate it to 20 trees, that'd be about 4,500 spotted lanternflies in comparison to what we were looking at with the red maple. And then, of course, by the 4th instar, we were down to 57 spotted lanternflies, which would be about 380, very similar to 2019 in terms of the densities that we were looking at out there. But one thing you'll notice in this slide is even though we got down to low numbers of nymphs, later in the season when the adults started coming back in, we ended up with 12,300 total adults in 2019. They were pretty much distributed evenly between the two sexes. In 2020, we had about 10,000, and they were a little heavier on the female versus the male in terms of the sex ratio. But as you notice, we had fewer eggs in 2019, even though we had more adults than we had in 2020. And you can see those numbers. And again, we didn't measure egg masses on the Ailanthus, but we would have had about 20,000 adults on 20 trees compared to that. So about twice as many on those Ailanthus trees had we counted for 20 trees out there in that year. But this just gives you a sense of that. And so it's not really the magnitude of the numbers, but we're showing you these because as we fit models to it, we want you to be aware we had large numbers that those models would fit from. They weren't small numbers that we were using here to estimate the seasonal activity. This just gives you sort of a sense of what the area looked like where we were doing the study. It was a housing development in Wyomissing, Pennsylvania. The Brian Walsh, got it right the first time. That's my neighbor. Get those two mixed up. So we're just gonna get that straight. But at any way, the orange dots are where the red maple trees were located. And the red dots were the wooded area where the 3 Ailanthus trees were located. And that'll become important as we see some of the later data here that we're going to talk about in a little bit. If you look down here in this corner, that's sort of what the trees look like, sort of what it looked like down at the ground level. Here's the trap on the tree and our little sign that says this Penn State study looking at spotted lanternfly. Okay, so this is probably the most complex slide we're going to talk about today, but this is where I want to describe what the difference is between the transition rate from one stage to the next versus what is the quantity that's in that stage at any instant in time, and what we're calling essentially, as we measured it, the activity period. So I'll explain that so you have a better idea what the difference between those two things are. So if we look at this, back, I guess there's a back here. If we look at this, these little cylinders that we have on here represent each life stage of the spotted lanternfly, from egg stage through the 4 nymphal instars to pre-reproductive adults, reproductive lay and fall eggs that then go through the winter that we're in right out there now. So as we start the season, everything's in this box. So all of them are eggs, right? So as we get enough heat in this over the course of time and we get enough degree days, these start to hatch and become first instars. And they do that at a specific rate based on what the heat requirements are for a population of spotted lanternfly. So this is the rate of spring egg hatch here. What's in this box here though is that new ones coming in here and minus any older ones going out, but once they get in there, they're going to be in there for a period of time because they have to have so much heat to go from being a first to a second instar. So that's a quantity measurement versus a rate measurement. And so what's in here is what we're going to be measuring at any degree day interval or on any day that we're out there sampling, is what's in the quantity in there at that point in time. And so that's all I really want to show you and kind of talk about here, because that'll become important as we move forward here. So one other thing with regard to that I did want to talk about here is that there's a little bit more to it than just as a transition from one stage to the other one. And so we have a number of trees. The number that's on that tree, whether it's on a degree day interval or a day of the year interval, is a number that's already in that stage. So at the beginning of the year, it's 0 if we're going into eggs, right? Plus any new individuals that just transitioned from egg to first instar, but also plus any that might have traveled from another tree or bush or something and came onto that tree. So you're getting immigrants coming in there as well. And then you're subtracting off individuals that are going to the next stage, plus any that might move off your tree to someplace else that's out there. And then of course, if they're dying, you're losing them as well, which I didn't include in that equation. But this is again a tree, new ones coming on, those leaving. But this is the thing that we're measuring as a quantity that's in that box at any point in time. Okay, so let's just look at this in a tabular form to give you a sense of what this might look like. So these are 25 degree day intervals crossed here. The transition rate, or the rate of movement from, say, eggs to first instar, and that's what this represents, would occur like this. So we're looking at 1,000, we're assuming there's 1,000 individuals this is how that 1,000 individuals would be distributed over the course of time, okay? And so you can see in that first box, even from 0 to 25-degree days, you might have 2/10 of out of 1,000 individuals in there. So that basically says if you had 10,000 individuals, 2 might hatch during that time interval, okay? But what you see here is the peak of that, where most of them are over here between 150 to about 275-degree days. So that's where most of the hatch is taking place out there. And that's the rate at which they're moving into that stage. The estimated activity period is what we measured in those boxes. Then we went back and fit a curve. We summed them all up, fit a curve to it, basically, and then figured what percentage of the total would be in there at any point in time, and then distributed those across here. But what you see is they might have some in there at the same time, but once you get out here, there's a delay or lag. So the peak of that is out in this time period versus back in here. And then the third line is just the activity period for the second instar, which is delayed over that. So if we look at 50% of the way through that time period, you're looking at about 210-degree days for the transition period, but you're looking at 270 for when you're going to have the peak activity taking place out in the field. And if we look at the second instar, then you're going to be out at 483-degree days when we hit that point. So you can see that there's about a 60-degree day difference here, but about 110-degree or 210-degree day difference there that's going on. And there'll be some rate of transition in the second along there too. But why is this important? And what I want to do here is just, I'm going to master this thing yet here. But I wanted to show you how this plays out in a cold year or a cooler year versus a warmer year at a given location. So if we look at this, 2020 was about 222-degree days cooler for the host season than was 2019, the 2 years we did this study. If you look at this, and you look at 50% egg hatch, that occurred on June 3rd in 2020, but it occurred on May 19th in 2015. So there's about 2 weeks difference there. So that's about a 14-day period. The peak activity was about 5 days later in both cases on June 8th versus June or May 24th. And then of course, we're way out here for getting to the second instar. So that shows it in a tabular form. What this does is shows you the same data, except in a cumulative curve, okay, over time. And this is what this would look like on a degree day scale. And so if this is degree days down here, what you're looking at is anything that's got a blue arrow pointing at it is the rate at which that happened on red maple. And red arrow is the rate it happened on Tree of Heaven, okay? And so what we're looking at is this year it was just 2020 data, but in the other years, this black curve is the average between 219 and 220 and how those models fit. And then the big broad bars are just the confidence limits on the whole thing, which are pretty broad in this case. But if you look at this, here's the activity period on red maple. Here's the activity period on Tree of Heaven. And one thing you'll notice is there's the one, even though they started similar time, it's stretched out further on red maple than it is on Tree of Heaven. Going over to second instar, you see here's the 2 different years on both sides there, and here's the fit to the 2 years combined. And so that's when it would happen for second instar. Here's Tree of Heaven over here. Again, you're getting that lag in there. Same way with third, fourth instar, and it just gets a little bit longer as we go on. So if you look at this lag period, starts out about 50-degree days difference, and it goes to about 100, maybe 150, 200, and maybe out here you get close to 300-degree days lag between the endpoint of those, depending on what tree species they're on. And then one last thing I did want to show is that if we look at that rate, remember, go back to that rate, what we're showing there, where that yellow line, is that yellow line on? I may have to hit this one more time, is Erica Smyer's model prediction of when spring egg hatch would occur relative to Tree of Heaven. Those are almost right on top of each other, right? But there's a little bit of a lag at the end on Tree of Heaven, but there's a big lag on red maple. So something's going on. They're migrating from one to the other, or there's a difference in development on those, or something's happening there that we need to think about. Okay, so if we look at those models for the nymphal stages, calculating degree days from January 1 to those events is pretty good estimate of when they're going to happen. We've got 2 years of data to show that we combined them and got a good fit to the model doing that. But when we get to the adult stages in egg mass deposition, the model doing January 1 didn't fit very well. And so this is just showing you a little bit about that. So this curve right here is the combined fit. This is 2020, 2019, what the model would look like in those individual years. Combine them, you get this fit that kind of tries to pull them both together and it splits itself across them. It doesn't. Here's for late season adult activity. Again, does same thing. And for egg masses, it's terrible. It just stretches the thing, and that wouldn't be a good predictor of when the eggs are going to be laid out there in the field if we do it from January 1st, okay? And so when we look at that, and again, I should have mentioned that that's early adult, late adult, and egg mass deposition that we're looking at. So we thought we needed a different place to start accumulating degree days, either make up some errors that build in there or some other cue that might be involved in when this is happening out there. So if we look down here, we went from accumulating from January 1 to accumulating them from when we saw the first adult out in the field and accumulated them. And when we did that, this is 2020, 2019, we see the combined fit is much better. Even for the early season, it's still a little bit of a spread, but it's a pretty good fit. But when we get to late season, when the females are showing up on the trees and they're occurring out there, we got a much, much tighter fit. We had an R square here of about 11, about 30 here. But when we got down here, it was like 98% fit. And when we looked at egg masses, again, there's a really good fit then under those conditions when we start with the first adult observation. So we're suggesting to use the first adult as your key to start accumulating degree days for the adult and the egg stage, but using January 1 for the nymphal stages, essentially. Okay, and this is just the same data, only just showing you the spread on a chart here for early adults, late adults, and then egg mass deposition at that location. This is just what we notice is there's 2 different dispersal time periods for spotted lanternfly. There's an early season dispersal, which is about 10 to 90% of that time period. You've got adults moving around for some reason at that point. And then over here, this is when the adult females are migrating and moving around just before they begin laying eggs out in the field. So there's a couple different periods that we need to be thinking about out there. So now I want to switch gears and talk a little bit about another cue that might be involved, and that's day length that maybe is driving some of these activities, behaviors of spotted lanternfly adults and when they lay their eggs out in the field. So this happened to be data from 2019 on red maple. And we were monitoring along. What we found early in the season was 80 to 90% of the population on red maples were male, and only 10 to 20% were female. But then for some reason, right around here, all of a sudden the percentage of males starts dropping off and the percentage of females goes up, and they almost cross and flip-flop. So by late season, 90% are females and only 10% are males. And so this is one of those things you were talking about. You see something with spotted lanternfly and you wonder why that happened there, but maybe it didn't, nobody noticed it before. So we thought we'd go back and look at another year. So we did it in 2020 again and got exactly that same pattern. Here's the male numbers up here, the female numbers, they crossed. But what's interesting in those curves is there's 250 degree days difference in when those events happened between the 2 years. One was a cooler year, one was a warmer year. So what's going on? If it was just simply degree days, they should be closer together. So there's something else going on out there in the population. So just to show you what happened on the ailanthus trees, look like this. Here's the female numbers were really high on ailanthus. The male numbers were really low. So we had a lot of females on ailanthus, not very many on red maple that were nearby. All of a sudden we get this cross, actually right here. After that point, even though this is jagging around there a little bit, we hardly had any spotted lanternfly left on the ailanthus trees, but we had a lot of them on the red maple. So our conclusion is, or our thoughts are, that they may be migrating off of there to the red maples for some reason at that point in time and leaving the ailanthus trees. And this was just before egg laying started out in the field. So if we put that all together, what we see here is that if we put the fall equinox on this graph, these are the 2 years when that occurred. It occurred at 1711 degree days in 2019 and at 1498 in 2020. So again, you see here's the females, or I mean the male numbers kind of tootling along here. Fall equinox view. 2020, males tootle along here, fall equinox view. So something may be cueing them as far as day length or something else that's correlated to day length that all of a sudden we're getting through that. So I need to hurry. I just got told I'm way behind here. So, so anyway, but down here we put it on a calendar date basis. Here's the fall equinox. You see they fall right on top of each other. So we think that the fall equinox is somehow involved in, or at least day length is involved in it. This actually shows Julie's data that in terms of the expansion of the female's abdomen, how big the yellow lateral line is in terms of area on here. But again, eggs, adult females, fall equinox, expanding at abdomens, and things are going on at that point in time. I'm just going to skip this slide here, I think, because of time and just try to get through a few things. But I want to quickly show you here what the topography has to do with everything. This is basically a topographic map of the Northeast United States. We have coastal plain. You can see that the elevation similar up the Hudson River Valley to the coast. Here's where the Erie Canal runs through the Mohawk Valley. You got your mountains, the Adirondacks, Catskills, Allegheny Plateau, Appalachian Ridge Valley, Green, White Mountains. You can see how that topography lays around here in this area. This is how the degree days fall. They track that topography. So you have a lot more degree days going up the Hudson River Valley, similar to what you got on a coastal plain, across where Erie Canal runs through, along Great Lakes, and so on. But you can also see that we don't have very many at these higher elevations, even way down into North Carolina. If you want to look at what we would project in terms of spotted lanternfly on where they might and might not occur, anything that's dark green, it says 100% of them can get from egg to adult stage in those locations. But if it's kind of this cream or a little bit below that, less than 1% could get there. So in those higher elevations, more northern areas, you're unlikely to have enough time for them to get the adult and then have time to lay eggs and do all the things they need to do. But just about everywhere else they can do that. Here's what a map of the US would look like relative to that. And then just to conclude, I'm going to skip this one too, but it's kind of interesting as well. But at any rate, just some thoughts that I wanted to leave you with here is the activity periods for spotted lanternfly is slightly longer on red maple than it is on tree of heaven. And that increases as we go across the life stages. If we accumulate from January 1, that works well for the nymphs, but it doesn't work well for the adults, which we need to reset that to first adult observations out there in the field. Day length may be involved in some of the timing of those late season things and the activities such as mating, egg or female movement behavior, and then egg laying that's going on. And the more northern areas, we're not going to have the likelihood of having spotted lanternfly. As far as some research questions are concerned, we want to know, do they really develop faster on tree of heaven or red maple, or are they moving, or are they doing both? And so that's something that we might want to look at. Environmental cues seem to be driving adult feeding and mating behavior, potentially as far as eggs are being laid as well. And then we want to ask the question, why do males and females appear, at least in our study, to segregate early and then remix later to mate, lay their eggs, and do the things they're doing? And then how long in terms of degree days is required once a female comes out for her eggs to be ready to lay? Can she shorten that, lengthen it, depending on these other environmental cues or what's going on out there? And then can the time from adult emergence to egg laying be shorter in areas with shorter seasons? Can they do that? Is it going to be longer and longer season areas? So those are good questions to ask. And why is there such a long time? There's about 2 months from the first egg to the first, from the first adult, the first eggs we see out there, regardless whether we're in Pennsylvania or whether we're in Virginia. So it's about 64 days. And then why is there such a long period of time? And then is day length the environmental cue that's causing this, or is there something else going on out there that we need to know? So just to conclude here and pull it all back together, why is this important? What's the value of this? Well, having these models better allow us to better time key activity periods and improve our monitoring, our surveillance, and our management activities that are going on out there. And then better timing of these management activities make our workforce more efficient, and they make our management more efficient as well out there and reduce our cost. It's going to help our pest managers or regulators determine where and when to do that. So it's going to happen at different times in different geographic areas, depending on degree days. So it might be 2 or 3 weeks difference from one site to another, maybe even 5 weeks difference of when you need to be doing these things. And then secondly, showing where females are likely to establish, or where spotted lanternfly are likely to establish their populations can be important, and knowing we don't need to spend a lot of time there. And then of course, we just showed you some future ideas for research. And so with that, I hope I didn't go over too long and really throw everybody else for the rest of the day behind here, which is the thing you get to do when you're first. But thank you for the opportunity to come and talk. And I really appreciate seeing some of you I haven't had a chance to see in a while. So hopefully this will be helpful information to all of you as you move forward with spotted lanternfly activities. And we'll talk to you later. Yeah.
Thank you. So the steepness of the crossing curves
Yeah.
would seem to indicate to me a really short season of actual mating. Is that the correct interpretation there?
I think I think that was on a degree day scale. So one of the things that doesn't show, it kind of truncates everything, that actually, even though some of those things look longer in early
Every invasive species can be thought of as passing through 3 different stages of an invasion process: transport stage, establishment stage, and then impact stage. And this all comes from invasion process theory. And so if you have transport, establishment, and impact potentials all being high for a particular species, then there's a severe risk of this species then essentially spreading and causing economic or ecosystem damage. And so using that framework, we're going to try and understand spotted lanternfly. And I'll talk to you how our team at Temple University uses this framework to try and understand spotted lanternfly. So we're using the invasion process to then develop all of our work. But of course, this is a team. And so I do most of the data science work, or I lead most of the data science work. We also have Dr. Jocelyn Bem, who does the field biology work, and then Dr. Benny Seibold, who does mathematics, who's here in the audience today. So he does the very hardcore mathematics that I'll show you, but we don't really do anything. It's really just them. We just complain a lot. And so it's really a lot of postdocs and undergraduates and a tech even, and of course, graduate students who are doing this work. And so I want to make sure that I'm clear with that. One of the first things that we did as a team was try to develop at least an internal framework for how we develop stakeholder apps under this invasion process theory. And this then allows us to essentially communicate our research results and the research results of others to the public and to stakeholders really, really rapidly. And so we have a website out where you can take a look at all those different apps. And so I'll be going through those different apps periodically throughout the talk. So first, this idea of transport. So where is spotted lanternfly transported? That's just a very general question that we all have. But that question, of course, requires a whole lot of data. And so lanternfly, this is a species of really high concern. And so many state and federal agencies then are essentially surveying for it. And because everyone does the exact same thing. It's easy. No, that's not true. Every single state and every single organization does things slightly differently. And so, that can actually, take time then to process. And so what we did was we developed a framework then for how we compile big data, such as what you get for spotted lanternfly, in order to understand invasive species spread. And so it starts off with first collaborating with agencies and also the public in some cases in order to collect data. And then we process those data that come from different sources, and that is not easy. Even just one data set can take you, weeks at a time to actually figure out, depending on how it is given to you. And then it goes through a very strict anonymization process. And so we round coordinates, and then if we are given any identifiers, we strip those identifiers out, et cetera. And then those data then are distributed as open access. And so this is all online. Everyone in your team can download these data. And so it's data map summaries, and then we coded it into our package, but you don't necessarily need to know R in order to use it. You can just download data and actually use it. And so then it produces essentially figures like this. And so currently we are almost completely updated for 2023, but it has over 800,000 spotted lanternfly records. And so we'll soon be over, 1 million records that are in the actual database. And so it produces images such as this one on establishment age. So how long ago was a particular site actually then lanternfly was found at? And please, for those of you in the audience, many of you already are. Contributing data is really easy. We'll take it whatever form you've got, we'll work with you. And more data clearly means better risk models, better able to test these risk models for your state and for all of us, the US and also the globe. And so then we developed different apps then to visualize this. There are many apps out there. And so this is one of the ones where you can sort of zoom in and take a look at the invasion progression, both at the county scale and also at slightly finer spatial scales as well. And so what we do is we merge and we think about interdisciplinary research a lot. And so the field biologists in our crew then essentially work with the data scientists to develop a multi-scale framework in order to manage or survey forest pest eggs at properties. And of course, we use spotted lanternfly in this case. And so we took this establishment age layer, this data layer that we smoothed out and did some other things with. And then we used it in order to actually do egg surveys to see what types of patterns we actually found, and then to sort of develop this multi-scale framework. And so for those of you who are in the field, it's intuitive and the data show this. And so this is something that we can verify. So at the invasion front, essentially, it's hard to find properties that have eggs. That makes sense, right? So these recently invaded areas, they have patchy colonization of properties that then makes these invaded properties difficult to find. But when you find a property, you're going to find eggs. There's lots and lots of eggs there. But in the invasion core and these older sites, then it's more difficult to find eggs. So you will definitely have eggs at your site, but it's difficult to find those trees with the eggs on it. And so we see this pattern across at least the invasion area here in Pennsylvania and around in the other states around Pennsylvania. So what properties do have spotted lanternfly eggs? I did not take any of these pictures. These are some of these pictures some of you actually took. And so it's the eggs that are probably moved the longest distances out of these transportation hub properties. And so probably eggs from China were imported in landscaping rock, maybe around 2012. I think people have more or less that idea. And please correct me if I'm wrong. Eggs are laid on stone, wood, metal, train cars, shipping containers, pallets, those sorts of things. And so any sort of property that's essentially moving this stuff, then is certainly what has a higher potential of having eggs. And this then means that it has a higher potential of these jump dispersal events. And so this is this accidental long distance transport, which essentially accelerates spread. And we can see that in the data. And so here is that 2014. This is first when they found it. And then they called Julie Urban. And they're like, come and save us. But Julie didn't save us. And so we have then 2015, then that has been spread. This is a joke, of course. The 2015, this was more delimited. Pennsylvania did a really great job at really trying to delimit this population. So the population was essentially this in 2015. Okay. In 2016, it did spread, but still we're centered there, in those counties. And then by 2017, that's when we're starting to see our first significant jump dispersal events. Why do I say significant? That's because we developed a framework to estimate what a jump dispersal event is significant or not. And so essentially what you do is you identify jumps with gaps in the distribution of distances to an introduction site that you can choose that's not essentially explained by a diffusive spread. That's what it is. And you have to do some complicated things because the spread isn't homogeneous, it's heterogeneous and that sort of thing. And so that then allows us to identify both isolated properties that have a jump dispersal event likely, and also clusters of properties, like you haven't gotten there, maybe it's been there a couple years ago, that sort of thing. So you can see here, this is when it spread into Virginia, New Jersey. This is that Pittsburgh population. I'm zooming out. There we've got it in New York and Ohio, Delaware, Maryland. This is when we get that one in Indiana. And of course, this is 2022. And then I don't have the 2023 data, but a lot of you know where it is now after that. And so in each case, then we can take a look at these jump dispersal events and ask what types of properties are these different jump dispersal events found at. And then that then allows us to then build apps that can map these invasion hub properties. And so we've done a lot of work trying to think about how we communicate these apps to the public. And so we've developed a framework for that as well. And so first what we do is we identify the types of hubs. Maybe we can do it through a really complicated statistical analysis, or maybe we just talk to all of you and you will tell us. And that's essentially what we did at the first, because all of you know whether you're seeing these different spotted lanternfly jump dispersal events. You attain these hub location data. You then have to clean and harmonize the data because all the data is always really different. Then build an online database that you map. You then think a lot about how you communicate to the public and the stakeholders, and you build that user interface. You deploy that application, and then you ask the states and ask the stakeholders, whoever is going to use it, if it's good or not, and then you do it again. And that's essentially sort of that repeat. And so we're building these apps at the state scale, and increasingly we want to build these apps at the smaller scale, say at the county level, because we can do a lot more of fine scale layering there at the county level. And so this is Erie, Pennsylvania, and then you can layer, different indications of whether there's a transport risk or establishment risk. Surveyors then can take a look at these maps on their phone and then essentially get driving directions to where they want to go and actually survey that particular location. And so you're going to use these apps then. This is how you're utilizing these risk apps. You're going to use these apps then to find these properties likely to spread spotted lanternfly. You can add different various layers. We developed the app in collaboration as much as possible with the states or with the user. So if you want something in particular map, then we'll try and do that for you as best we can, or you can give us data to map as well. And so about 17 maps are built. And so if you're a state that doesn't have one of these apps, contact me right away, and we will work to get you an app right away, okay? And so I've talked now about 4 different frameworks: one to compile big data, one to manage forest pests, eggs, one to estimate jump dispersal events, and one then to map these invasion hub properties. So let's move on to establishment and growth. And so one of the most recent frameworks that we're starting to work on, and this is driven by a student of mine, Sam Owens, is trying to understand how climate change is impacting our invasion risk assessments with spotted lanternfly. And so we're essentially building these establishment models based on both the invaded and the native ranges. And then we're assembling these predictions with high transport and high impact potential properties. And so this is a statistical niche model. This is not based on going out and sampling like what Dr. Calvin talked to us about before. But this is on then using the records that are in the public sphere and trying to then build huge, essentially machine learning correlative type methods then. And so we built these different models then based on subsetting these records. For various reasons, it's good to subset the records to try and understand what these data are telling you. So we built one on all the records across the globe. We built one just for China, which is that native area. And then we built one only for the US. And so I'm gonna talk you through these and show you what I hope is sort of the main message of what these apps are telling us, what these maps are telling us. So essentially, with that contemporary climate that we have now, this is all the data, this is that global data. All those different points there are these important viticultural growing regions in the United States here. And essentially, you see sort of this pattern where, it can establish in a wide swath of the United States, but that mountainous west region is sort of iffy. You can see that there. And then this is what you get in future climate. Here, you see that shifting up. For example, we actually see that contraction in the south. If you take a look at that, it's not just further north, but you see that whole range moving. So this is just one sort of estimate of what's going to happen based on all the global data. If you know talks, there's always a catch, because what's the next model going to show? Okay, so then this is just built on the native range data. Now, this is important here because we did everything we could in everything. The student is amazing. He did stuff that I don't even understand to try and reduce the effect of there not being great samples in the native area. So he really tried to deal with that issue. But certainly there needs to be better samples in the native area. But by just building that app, by building that map, then you can see the vast difference here in this range. Actually, starting to match, some of those developmental models, that Dennis just showed. That sort of thing. And you don't see that contraction, you just see an expansion then, at least for native range. And if you think about it, and for those of you who sort of have looked at these data, I mean, there are lanternfly that are found in the tropical location, spotted lanternfly found in tropical location in China. And they're found really, really far north too. And you can see these areas, and I know people have seen lanternfly in the tropics of China. But this is that invasive range. This is just the US. Do not use this model. This is not true, okay? But what it really illustrates is just the small amount of climate that lanternfly has currently invaded. It's a very, very small amount of climate because this is our best estimate, and that's not even true. Of course, you had that shifting that affects it as well. And so we are going to see lanternfly spread is sort of the take-home message here, and we don't necessarily know exactly what the best model is, but somewhere in between all these different models that we're looking at here. And so those models then, maybe will be underestimating, spotted lanternfly range potentially. From these statistical approaches that we're looking at. So compared to a statistical approach, then you can have these first principle models. And this is where the mathematicians come in. And these are mathematicians that do stuff that I do not understand. They are very, very good at what they do. And so we're taking then the work that many of you are doing, Dennis Calvin's paper that he just talked about, that is the backbone development model behind this. And then they've put together a system of partial differential equations in order to model the population dynamics of spotted lanternfly. And so one of the first things they did was they developed this really novel moving mesh numerical method. It essentially allows you to solve for the growth rate of a pest population dynamic model based on mean seasonal profiles. That doesn't mean a whole lot, but it means that what we can do is we can start aggregating tons and tons of temperature data and come up with really rapid estimates as to growth rate or establishment potential. That's the idea here behind having these first principle models. And so you essentially look at these seasonal patterns, these annual patterns. You have a mean temperature across the year, and you have some sort of amplitude, and then those you can solve for R naught, essentially solve for the growth rate. That's what you can do. And so then you can build an establishment map based on this. This is parameterized based on some of the older studies that are out there. And so I think at this point, once we reparameterize it, it's going to be a little bit further north if you look at that. And so this population growth, this model essentially is built on this type of understanding of thermal biology, of physiology, is that there's some type of thermal performance curve and you have a population growth rate on the y-axis and then temperature, and there's some sort of Tmax. There's that maximum growing degree days, right, for development that you've got. But there are multiple vital rates that are going into this sort of very simple idea. We talked about development just in the previous talk a whole lot. We have fecundity, mortality across all these different life stages, and they all have very different looking functional forms potentially from the data that are out there. So this here is our best guess as to what these vital rate functions look like based on very scant data. This is our best guess essentially. And so what we did was we extracted a lot of published data on this temperature dependent vital rate data then in order to build these. And I want to just really explain to you sort of what goes into this and decisions that have to be made and how more data needs to be done, more experimental data, in order to really get these vital rate functions correct, because they can have vast differences. So that's our vital rate equation for egg mortality. It's based on data like this. There's just 4 data points there, but I drew a line between them. You can sort of see that curve. There's 4 data points. I can fit anything to this, right? It's 4 data points. And so, okay, well, maybe that double breakpoint looks the best, or what does it actually look like? But just with those simple changes, it really vastly changes that spread. And so whenever you have those critical transition points, such as when you start to have egg mortality when it gets too cold, that could really then determine where your estimates are going to be. And so that's really where we need to focus our experimental work, I believe, so we're at those transition points. But all of this work so far has really been focusing on mean temperature differences. But we know that with climate change and just reality, the climate varies a whole lot throughout the year. You have climatic variability that occurs. And so we made a framework to estimate this effect of climatic variability on establishment and growth. So essentially, we're comparing then model simulations with the mean temperature sinusoidal curves to model simulations with something that's jagged, that has daily temperature fluctuations. And we can increase that noise or decrease that noise and essentially look at how this variability affects our population growth rate estimates. So we can simulate that growth rate across sites all across the United States using temperature data that we can get. Just going to focus on two, which might be interesting for many reasons, but they're pretty close to each other and they're on the West Coast, Napa and Yosemite. So what I have here on the y-axis then is the simulated growth rate. We've done about 1,000 simulations in this figure I'm going to show you. And on the x-axis then, I'm increasing that noise. I'm increasing that variability is what I'm doing. There's going to be two lines here. The first line, the top line then, that's the R0 value. That's the growth rate of the mean seasonal temperature without any variation at all. And then the other one is just at 1. That's essentially where populations either grow or decline. And so essentially increased variability, that red area, that's higher growth. Anything above that line, below that line means there's lower growth rate estimated with variability. Anything below 1 then means a declining population. And so this is Yosemite here. Variation clearly, on average, essentially causes populations to decline. You get a little bit of increase in growth, but not really through all. You get it pretty increasing. But this is Napa. And so here you can see a major difference, and it has to do with all that complicated interactions among the vital rate functions, and then essentially that temperature profile in Napa versus Yosemite. Very different results. Here you can have certain years where the variability itself causes 2 times the amount of growth rate. Sure, it can cause the other direction, but it causes 2 times the amount of growth rate. And it takes more variability to actually push that population, then push those populations to have it be a declining population. And so we can do that across the United States and essentially highlight counties where, if everything else is great for lanternfly to grow, where this climatic variability might make lanternfly kind of sticky. It's going to be hard to get rid of them, sort of that idea. It's where you might get these outbreaks occurring. And so I told you about 3 more frameworks here. One about how climate change increases variation, one numerical method in order to how to solve for growth rate, and then one that includes climatic variability. Okay, so let's finally talk about one final one, impact. I don't do any of this justice. I just sometimes talk to vineyard operators, and these are some of the quotes that I've gotten from them. And so clearly, spotted lanternfly impacts grapes in Pennsylvania. During the pandemic, we had a lot of time on our hands, which isn't true. We just thought a lot about pandemics. And us, as well as other people in the field of invasion biology, essentially realized that the invasion process is analogous to the infection process. So if you think about transport, the same as transmissibility, establishments, the same as infectivity. Typically, transmissibility and infectivity are combined. And then, of course, impact is essentially virulence, whether or not you get sick or perish. And the CDC has developed lots and lots of different frameworks, and they developed one called the Pandemic Severity Assessment Framework. It's really nice. It has a nice intuitive numbered scale, has intuitive quadrant plots. It's a way to take really complicated scientific ideas and translate it into something that's meaningful then to the general public. And so what my graduate student and I did was we essentially came up with a way to try and adapt that to lanternfly. And so we first had to define what we thought of in terms of saying this term, pan-invasive species. So this is a regional pest that becomes a globally invasive species that we all know about, but that globally invasive species essentially starts to impact global markets, just like what a pandemic would do. And that's essentially what we define pan-invasive species. And so we took that pandemic severity assessment framework, and then I'm not going to go through all of this, and then we adapted it to essentially invasion process theory to come up with this severity assessment for invasive species. So this required us then to essentially build these establishment maps. Think about transportation probability. So those gray lines there, those gray arrows are states that import a whole lot of products from the invaded states. And then, of course, where there are these grape growing regions, these important grape growing regions. We did that both in the United States and also here. You can see a lot of those lines heading over to where we import a lot of wine, as you can see. So a lot of that transport risk is there. You combine all that, you do all that, and you can come up then with a scale between 1 and 10 in terms of whether or not spotted lanternfly has a severe risk of spreading to impact the entire global market of wine, and it's an 8 out of 10. And so this is just sort of a very simple measure that you can give. And you can see that here if you look on the y-axis predicted invasion severity, and then if you go up to essentially that wine export market size, there you can see Spain and France and Italy those types of areas. And then there's been other models that have also been developed that have essentially shown that, yes, in that area of Western Europe is where lanternfly might spread to next, most likely. And so we've also then developed a risk gap to zoom in on the global scale and take a look at all these different layers that we've produced. So why did I just go on about all these different frameworks really quickly to all of you? So I'm going to take it back and then end my talk here. So why did we develop these based on the invasion process? So we believe that the invasion process theory essentially provides an intuitive way to partition these complexities and complications of invasive species spread in a way that can be easily communicated then to the public and amongst each other, amongst researchers, et cetera. And then frameworks are general, and they can be adapted then to any species early or late in the invasion process. And so Dana said, if we only knew then what we know today, well, now with these frameworks, we could know today for the next spotted lanternfly that's coming, and hopefully we can then adapt these relatively quickly. And then finally, these apps allow for essentially this rapid and effective communication of research to stakeholders and to the public. And so this then enhances our ability to rapidly respond. Whenever we find something new in research, if we're building these apps, then this is much easier for stakeholders, for those of you who are dealing with things in the field, then get your hands on the latest research. Okay, that's it. So there's my website there. There's publications for all those publications and the apps you can see there. Thank you so much for listening.
Yeah, that's really great. I think you guys have really come a long way since I was,
Yeah.
before I retired anyway, so you're making good headway there. But one of the things that you might want to think about in the future too, as you develop further on your models, is when we work with the European Corn Board, one of the things that becomes real important is that climate comes into it, is the timing at which the pest occurs relative to the host. And they can be put in and out of synchrony based on whether you have a warm year, cold year, whatever it could be. And of course, with corn borer, we had different planting dates. So we had plants at different stages of development, and they would go into cornfields based on the stage of development. So depending on which ones were at the right stage versus not could change the impact that they had, because the timing became critical to how much damage they caused to the plant as well, and yield losses and stuff. So as you move further along, that may be something you want to look at. And the other thing I was going to say is in Pennsylvania, we also simulated year by year the climate for 30 years with European corn borer. And what we found was that in the warmest areas of the state versus the coldest area, which was Potter County, the warmest was York, there were 5 weeks difference when all these events happened. And then within a year at the same location, there was about 5 weeks difference between the coldest year versus the warmest year. So that variability you're looking at in climate can really play out in some really interesting ways, I think, as you look at some of these pests.
Yeah, thank you. I was wondering with your talk even, how much did variability drive that, between year difference? Certainly the mean was different, but was that colder year more variable too? Like what's sort of going on there?
Yeah, that's a good question. I didn't look at that, but that would be a good question too, which would affect things. So, yeah.
One more if there's time.
Nice talk, Matt. I was wondering, I think I've asked this before, but I'm wondering how your thinking has gone on in terms of the role of humidity in kind of these predictions, especially towards California, et cetera.
Yeah, so with the niche models, that's where we account for the precipitation, is in these statistical niche models. And so we've not necessarily humidity, but precipitation and other variables, remote sense variables that we can do based on water content, essentially. The first principles models, we don't have that yet. And so again, I think that's another key area where we need to be stressing out, say, grapevines or stressing out plants, and then seeing how that affects, mortality, development, fecundity, those sorts of things. And so I think that's where we need a lot of the research. Yeah. Yeah. Yeah, so our mathematician has just said, we know how to put it in. It's just then we've got that framework in place. It's just getting those data then. Mm, mm, mm. Yeah. Okay, thank you so much.
Okay, the next presentation is going to be virtual. It's Melody Keena from the U.S. Forest Service.
All right, sorry I can't be there with you today. We didn't have travel funds to allow this, but hopefully this talk will still be easy for you to see. I have assigned a slightly more specific title to it, Heat Treatments to Kill Eggs of 2 Invasive Insects. I'm going to talk also about spongy moth because we're doing both insects in this study. This came out of a novel idea that a conversation with Mike Gallagher, one of my co-authors on this, where we thought that it might be worth looking at prescribed fire to see if that would have an impact on invasive insect eggs that would be at least lower on the tree boles. And so that's where this started from. And Mike is a prescribed fire scientist. And we also brought on Nushin, who is a postdoc through the West Virginia University, and Tony Cohen is with APHIS. He's been involved in kitting egg masses and finding sites and things like that. All right, so the spotted lanternfly populations that we used for these studies, we collected from a wide range of populations. As far north as Massachusetts and as far south as North Carolina. We wanted to incorporate as much genetic variability as possible in what we were using so that we could see if there was any impact of that as well on the responses to these temperatures. These were collected October or the first week of November in 2022. And we collected the eggs by removing small pieces of bark with the egg mass on them. And then we took as much bark off from around the edges as we could. And we dried these eggs under a hood for 30 minutes just to get the bark so it didn't contain moisture and would grow fungus. This prevents fungal growth through the remainder of the treatments. We also used spongy moth laboratory colonies, one from Connecticut and one from far east of Russia. So it's an Asian strain as well as a North American strain. And this insect also travels on all kinds of commodities, just like spotted lanternfly does. And for these, we grew the larvae on artificial diet, mated them, had the females lay on paper, and then cut the egg masses longitudinally so that we could spread out the eggs that we produced. There's probably about 1,000 to 2,000 eggs in one of these egg masses because these are large females, so that's a lot to count. We needed to spread that around. So all of these egg masses were held in an alternating temperature regime that was mimicking Scranton, Pennsylvania. This temperature regime on a weekly basis, the maximum minimum temperatures changed and then it ramped between those two temperatures during the day. We also changed humidity and light cycle to mimic that location. The egg masses were kept in this treatment until March, which was the timing of the field work with the prescribed fire, and then they were exposed to different temperatures to see what would happen. Under this regime, SLF hatches in that green window that you see there. It starts hatching really late in May, and most of the hatch occurs in June and spreading on through beginning of July at that temperature regime. And these were the treatments that we applied to the insects. We didn't have as many spotted lanternfly egg masses as we wanted, so we couldn't do as many treatments or have as many egg masses per treatment as we would have liked. But we did place them directly into this regime, whereas the spongy moth eggs, we gave 30 days at 25 degrees to make sure they had embryonated before we put them in the alternating regime. And our temperature treatments were from 35 degrees Celsius to 70 degrees Celsius for 15, 30-minute, 60-minute, or 120-minute intervals. These treatments were done by pinning these egg masses to balsa wood, and then these plates of egg masses were placed in an isothermal chamber, and the chamber was brought to the temperature desired for the length of time desired. And this just gives you an idea of what happens for this type of chamber. It would happen in a commercial facility too if you were running these type of temperatures. You open the door, the temperature drops, and then you have to monitor how long it takes to get to the temperature you desire. So for example, here I'm showing 60 degrees and 70 degrees. 60 would be 140 Fahrenheit, 70 would be 158 Fahrenheit. So the doors opened, it takes, a few minutes. In these cases, it was taking about, let's see, 14, 15 minutes to reach the temperature. We would start the timer when the temperature was 1 degree below the set point that we desired, and then continue to monitor how long it took to reach the actual set point. Once the eggs were treated, spongy moth eggs require some extra humidity to hatch properly. So we put them in these boxes with a grate in the bottom and some water under the grate to keep the humidity high. Spotted lanternfly isn't as sensitive to humidity. This chamber was 25 degrees C and 60% humidity. And so they were held at that condition rather than being put back in the alternating regime, which I think may have caused some issues, but I'll explain that in a little bit. As the eggs began to hatch, we would daily remove all the hatch and count it. This was several technicians working 7 days a week, 8 hours a day, massive study. Once the eggs had all hatched and no further hatch was occurring, lanternfly eggs the fluff was brushed off and all the eggs were counted. For spongy moth eggs, you have to get rid of all the hair, and then you have to count embryonated and unembryonated eggs that didn't hatch. And I'm just showing some where most of the eggs had hatched. You can see the unembryonated are the orange color and the embryonated are the darker colored ones. But for egg masses where nobody hatched, this could be 500-600 eggs per half egg mass. So here's the spotted lanternfly for this preliminary study. The egg hatch wasn't what we had desired. It was only average about 50% for the controls. And so I've drawn the mean control hatch line on here in red so you can see where that falls. We started seeing reduced hatch at the highest time frame for 45 degrees Celsius. And then at 50 degrees Celsius, 15 and 30, there's also reduced hatch. And the highest temperature at which we saw any hatch, and it was, I believe, only 1 or 2 individuals, was 55 degrees Celsius. So 50 degrees Celsius is 120 degrees Fahrenheit. And most of the eggs will not survive for 30 minutes at that temperature. We have to do more on this because we have to get more egg masses into the study. And we also have to narrow down to this range of temperatures at which the mortality is occurring. Here's a better example of what we had hoped to see. This is the spongy moth results. 95% or higher hatch in the controls. You can see that at 50 degrees Celsius is where you start seeing eggs not hatching, and then when you get to 55, it was a couple of egg masses that had a few individuals hatch, but it was for only 15 degree treatment. Again, that's only 131 degrees Fahrenheit. 50 is 122, just for those that don't translate between the two well. And here's just some pictures of the prescribed fire. These were very low level prescribed fires. We had egg masses at 0.5 meter, 1 meter and 2 meters. And the egg mass there on the left-hand side is one at the 0.5 meter that got royally toasted. For this situation, we had 3 different sites. Very few egg masses were out there for this preliminary study, but at 1/2 meter, almost all of them were killed. And even at the one site at the Silas Little Experimental Forest, the 1 meter, most of the eggs were also killed. Future work is going to work with a little bit higher intensity prescribed fires and additional egg masses, and also look at the spongy moth. We had 4 egg masses to use in the field for the first year. For this year that we're currently in, we have collected a lot more egg masses, and thanks to those that have shipped some additional egg masses. We have already treated a fall set of eggs. First year we only did spring eggs. We've treated a fall set of eggs over this range of temperatures. We're looking at primarily 45, 50, 55, and 60 degrees Celsius for this year. And we will treat also the same temperatures, but a couple different other intervals for the spring eggs. The other thing that we did this year is we made sure that all of these eggs had 14 days of 20 degrees C before putting them in the alternating regime or treating them, to ensure that they had reached the point that they would be impregnated enough to enter diapause if they were going to. And for spongy moth, we're doing the same thing, fall and spring, but we're concentrating just on that 50 degrees to get a dose response curve for both the Connecticut and Russian strains. So what we did see, if you remember those box plots that I showed you, there's substantial variation between egg masses and percent hatch across all the treatments, but particularly where you started seeing mortality occurring. And spongy moth eggs hatch sooner than the spotted lanternfly eggs, which is an interesting finding alone. Spotted lanternfly eggs didn't hatch well even in the controls, probably because not all the eggs had entered and could complete diapause normally under the conditions we gave them, both not allowing them to impregnate first and also moving them from the alternating regime to a 25-degree regime for hatch instead of putting them back into a normal regime for hatch. Percent hatch of spongy moth eggs showed decline at 50 degrees C for all time frames. And for lanternfly, we were starting to see some mortality occurring at 45 degrees C with longer treatments, but all 50-degree time frames showed some substantial mortality, and 55 seemed to be the temperature at which most eggs of both species died, which is interesting. The temperatures required to kill eggs for these species are similar to the ISPM 15 standard used for killing wood-boring insects and solid wood packing material. ISPM 15 protocols require core temperature of the timber to be heated to 56 degrees Celsius, which is 133 Fahrenheit, for a minimum of 30 minutes. And if you use that treatment, it would be sufficient to kill the eggs of both of these species, since we were seeing mortality above 30 minutes at 55 being complete for both insects. And this work took a lot of technical staff, a lot of people helping with the egg mass collections, and this work is being funded by the bipartisan infrastructure legislation through an agreement with West Virginia University and by Forest Service funding. And I will stop talking if there are any questions. Feel free to email me if you have questions. It's just melody.keena@usda.gov. Thank you for listening.